Hi, my name is Meg, and I'm going to provide today in this video a brief introduction to hyperspectral imaging. Hyperspectral images represent both the spatial and spectral image characteristics of an object when an object is imaged at different wavelengths. As you can see here with the retina, it has been imaged at a variety of wavelengths in the visible and near infrared portion of the electromagnetic spectrum. We can find spectral and structural signatures related to specific elements, chemicals, or chromatic properties. And this has many applications in industrial inspection, food processing, or as you can see here, in biomedical imaging of the eye. In the case of the eye, we can image it at different wavelengths by emitting a light source directed directly at the retina, and then collecting the partial reflectance and transmittance by the retina using charge-coupled devices. Now what we see is that we collect both spatial information in the form of rows and columns for each wavelength, but also a series of bands for the spectra itself to produce a pixel spectra across all of those bands. Typically, we find that the hyperspectral image sensors have a high spectral resolution, as you can see here, and a low spatial resolution. For the clinical application of imaging the retina, we can take slices of the eye and analyze them for the detection of disease. Specifically, how retinal tissue reflects light at different wavelengths can be a physiological indicator. For instance, amyloid beta is a biomarker for Alzheimer's disease because it accumulates over time in the retinal pigment epithelium, or RPE, and is correlated with brain amyloid beta levels. Hadou et al. In 2019 found there was significant differences between control and transgenic mice with their amyloid beta levels between the retina and the brain. So this technique is very powerful for understanding certain diseases. How do we go about actually processing this hyperspectral image data? There are a few steps. We can collect the raw data, represent the data as a hypercube, perform pre-processing to denoise and sharpen the images, then perform this special step called spectral unmixing, and lastly, interpret the results. A hypercube is shown here, and it is a hypercube object that stores the data cube, spectral wavelengths, and metadata. And what you see is really it's a 3D array where you have the spatial dimensions of the acquired data, but also the spectral dimension specifying the number of wavelengths used during the acquisition. In MATLAB, we can see that we can define a wavelength range as well as the number of bands and create a linear space representation that then is fed into the inbuilt MATLAB function of hypercube to create our hypercube representation. Our next step is to pre-process, and we can do two general things. One is to remove some of the sensor noise or low resolution distortions through non-local denoising. There's an inbuilt function called denoise ngmeet in MATLAB that performs this. We can also perform pan sharpening that fuses hyperspectral and panchromatic data. And sharpen CMNF is a MATLAB function that helps us do that. The next step is to perform spectral unmixing. Spectral unmixing is needed because intensity values are recorded at each pixel and specify spectral characteristics of the region the pixel belongs to. And this can be broken down into two general surface types. One is homogeneous or pure pixels, and they're shown at the very top in blue and yellow. We can also have heterogeneous surfaces that are a combination of two or more homogeneous surfaces, and they're shown here in green as mixed pixels. We have several mixed pixels because of the low spatial resolution typically of the hyperspectral sensor. In order to actually perform spectral unmixing, we want to decompose those mixed pixels into their constituent end members or pure pixels. In the case here, we have two end members, the blue being end member 1 and the yellow being end member 2, and we want to break them down into a mapping. There's two steps that we do to do that. The first is end member extraction, and the second is an abundance map estimation. So in order to actually achieve this, we want to extract the prominent features of the end members, and there are several techniques, including the pixel purity index, to actually estimate the end members by projecting pixel spectra to an orthogonal space. and identifying the extremes. There are several other approaches, but given this step, we can then use abundance map estimation to actually estimate the fractional amount of each end member present in each pixel. Based on this, we can actually produce an abundance map for each end member, and that can label each pixel as belonging to one or the other of the end member spectra. To interpret the results, that's our next step, we can actually produce all the end member signatures plotted by band member and their relative amplitude, and we can produce those abundance maps. So here are examples for a landscape of different materials, such as asphalt or meadows or trees, and their abundances that are computed using a full constrained least squared approach. We can combine all of these together into one image so that we can see all the constituent materials present and actually quantify how much there is. So in summary, to perform hyperspectral image processing, we want to represent the signals as a data cube, perform pre-processing to clean and denoise the images as much as possible, unmix the mixed pixels so that we can actually compute the abundance maps and then interpret the results. So that is the end of this video. Thank you very much for watching.